Curing people is the whole point, right? But if there is one thing that the Human Genome Project has taught us, it's that finding cures is a whole lot harder than simply reading letters of DNA. Take, for example, the case of little Riley Dimanche.
At 2 months, Riley appears to be a perfectly healthy baby boy, but he's not. When Riley was just 13 days old, Kathy Dimanche got the call that every parent dreads.
My pediatrician called on a Thursday evening, and he said, I need to talk to you about the baby. He said, are you sitting down? And I'm like, yeah, and that really surprised me. And he said, are you holding the baby? Because he didn't want me to drop the baby, obviously. And he said that tests came through and Riley tested positive to cystic fibrosis. And I was in shock.
As Kathy and her husband would soon learn, cystic fibrosis, CF for short, attacks several organs of the body, but especially the lungs. Its victims suffer from chronic respiratory infections, and half of all CF patients die before the age of 30.
Sounds good.
Good.
Sounds great. But I think we can still be hopeful that their child will grow up to have a normal, healthy, happy, and long life. But at the present time, I don't have any guarantees about that.
Someone had asked me, are you prepared to bury your son at such a young age, whether it's 4 or 40? And he was 17 days old when that happened. And I said, I've had him for 17 days. I wouldn't trade those 17 days.
Finding the genetic defect that causes CF was big news back in 1989.
Medical researchers say they have discovered the gene which is responsible for cystic fibrosis, the most common inherited fatal disease in this country.
We're going to cure this disease.
A lot of people expected the cure to arrive any day. It didn't. Francis Collins, now head of the government's Genome Project, led one of the teams that discovered the CF gene.
We still have not seen this disease cured or even particularly benefited by all of this wonderful molecular biology. CF is still treated pretty much the way it was 10 years ago, but that is going to change.
The original hope was that babies like Riley could be cured by gene therapy, medicine that would provide a good working copy of a broken gene. But attempts at gene therapy have hardly ever worked. They remain highly controversial, so if there's going to be an effective treatment for Riley, instead of fixing his genes, we're going to take a look, and this is new territory, at his proteins.
What do proteins do? When you look at yourself in the mirror, you don't see DNA, you don't see RNA. You see proteins and the result of protein action. That's what we are physically composed of.
So it's not a Rodgers and Hammerstein thing where one guy does the tune and the other guy does the lyrics. This is a case where the genes create the proteins and the proteins create us.
That's right. We are the accumulation of our proteins and our protein activities.
A protein starts out as a long chain of different chemicals, amino acids, but unlike genes, proteins won't work in a straight line.
Genes are effectively one-dimensional. If you write down the sequence of A, C, G, and T, that's kind of what you need to know about that gene. But proteins are three-dimensional. They have to be, because we're three-dimensional and we're made of those proteins. Otherwise, we'd all sort of be linear, unimaginably weird creatures.
Here's part of a protein. Think of them as tangles of ribbon. They come in any number of different shapes. They can look like this, or like this, or this. The varieties are endless. But when it's created, every protein is told, here is your shape. And that shape defines what it does, tells all the other proteins what it does, and that's how they recognize each other when they hook up and do business. In the protein world, your shape is your destiny.
They have needs and reasons to want to be snuggled up against each other in a particular way. And actually, a particular amino acid sequence will almost always fold in a precise way.
Should I think origami-like? I mean, should I think folding and then...
It's very elegant, very complicated, and we still do not have the ability to precisely predict how that's going to work. But obviously it does work.
Except, of course, if something does go wrong. And that's what happened to baby Riley. Riley has a tiny error in his DNA. Just 3 letters out of 3 billion are missing. But because of that error, he has a faulty gene. And that faulty gene creates a faulty or misshapen protein. And just the slightest little changes in shape and boom, the consequences are huge. Because it is now misshapen, and a key protein that's found in lung cells, in fact in many cells, can't do its job. So let's take a look at some real lung cells. We'll travel in. This is the lining or the membrane of a lung cell, and here's how the protein is supposed to work. The top of your screen is the outside of a cell, the bottom of the screen the inside of the cell, of course, and our healthy protein is providing a kind of chute so that salt can enter and leave the cell. Those little green bubbles, that's salt. And as you see here, the salt is getting through. But if the protein is not the right shape, then it's not allowed into the membrane. It can't do its job. And without that protein, as you see here, salt gets trapped inside the cell, and that triggers a whole chain of reactions that makes the cell surface sticky and covered with thick mucus.
The first 2 positions that are done sitting up are probably a little more difficult to do.
That mucus has to be dislodged physically.
Okay, just kind of like that.
Riley's family is learning to loosen the mucus that may develop in his lungs and fight infections with antibiotics.
You want to sort of do it with a cupped hand.
Trying to get at the top of the lungs.
Yeah, you want to be like
But what the doctors and the scientists would love to do is if they can't fix baby Riley's genes, then maybe there's some way to treat Riley's misshapen protein and restore the original shape. Because if you could just get them shaped right, the protein should become instantly recognizable to other proteins and get back to business. But look at these things. How would we ever learn to properly fold wildly multidimensional proteins? It may be doable, but it won't be easy.
The Genome Project was a piece of cake compared to most other things because genetic information is linear. It goes in a simple line up and down the chromosome. Once you start talking about the 3-dimensional shapes into which protein change can fold and how they can stick to each other in many different ways to do things, or the ways in which cells can interact, like wiring up in your brain, you're not in a one-dimensional problem anymore. You're not in Kansas anymore.
And as scientists head into the world of proteins, they're looking very closely at patients like Tony Ramos. Tony has cystic fibrosis, but it's not the typical case. CF almost always develops in early childhood. Tony didn't have any symptoms till she was 15.
I started having a cough, and then we kept thinking I was catching a lot of colds. And my stepmother thought, that's not right, so I started going to doctors trying to figure it out. And went through a lot of tests because I don't fit the profile. Tuberculosis, walking pneumonia, test after test.
At the time of her diagnosis, Tony's family was told she might not survive beyond her 21st birthday. She's now in her mid-40s. But her CF is worsening. 2 or 3 times a year, she does have to be admitted to the hospital to clean out her lungs.
Temperamental. They were always doing some little funky study to help the cause because we're not the normal, there's not a whole lot of us. I know that they don't know why, and it's the big question mark, and hopefully, research will keep going and figure it out.
Here's the question. Tony was born with a mistake in the same gene as baby Riley. And yet, for some reason, when Tony was a baby, she didn't get sick. Why? And now that she is sick, she hasn't died. Why? What does Tony have that the other CF patients don't have? Dr. Craig Gerard believes the answer lies in her genes, in her DNA.
How are you?
Good.
So, do you think that the change in the antibiotics is helping you?
Yes.
Yeah.
And I dropped 4 pounds overnight.
That's a lot of weight.
Yeah.
Okay. Mind if I have a listen?
Nope.
No gene acts in isolation. It is always acting as a part of a larger picture. And there can therefore be other genes which compensate.
Could it be that Tony has some other genetic mutations, good mutations, that are producing good proteins that kept her healthy for 15 years and are keeping her alive right now?
You sound a lot better than you did when you came in. So I think you're on the mend. Okay, hang in there.
Thanks.
Bye.
Bye.
In my opinion, there are genes that are allowing her to have a more beneficial course, if you will, than another patient.
Good. Sound good. Okay.
Dr. Gerard is searching for the special ingredient in Tony. If it turns out she has 1 or 2 good proteins that are helping her, maybe we could bottle them and use them to help all CF patients like baby Riley.
Knowing that if there was ever an emergency and I didn't know how to do it and I couldn't get in touch with you.
No one can predict Riley's future or to what extent CF will affect his life. But now that we're getting the map of our genes, we'll be able to take the next big step. Because what genes do, basically, is they make proteins.